class. Moving on to chapter 3, molecules within the cell. And microbiology is one of those fields where it's pretty clear that the cell is only made up of molecules, and all the molecules then within the cell are very, very important. Okay. After this chapter, we should be able to draw the structure of an atom, describe the various simple sugars and polysaccharides, compare and contrast fats, lipids, phospholipids, and sterols, draw an amino acid and resulting 4 levels of protein structure, and identify parts of the nucleotide and distinguish between DNA and RNA. Okay, that is sort of a lot to summarize in one chapter, but it's what we need to be able to understand all the molecules that are important for microbiology. Okay, now one of the very first experiments was by Stanley Miller, and Stanley Miller did this interesting experiment where he made this primordial soup. So let me just, okay. He took certain chemicals and then boiled them and had them go up through this chamber that had an electrode in it. And so once you add electricity to this, you actually get a mixture of amino acids and some of the other building blocks needed for a cell. It's been sort of hard to reproduce this experiment, but this is what people think happened originally, that there were these chemical blocks that with electricity, with lightning in the atmosphere, we were able to make all the chemicals that we need to have a cell. Okay, is different by the number of protons in the atom. Now for biological chemistry, the major players we need to think about are hydrogen, carbon, and oxygen. Okay, almost every molecule that's inside of a cell is made up of carbon, oxygen, and hydrogen. Now, as we take these different elements, the different atoms, and put them together, then we can bond them together, and the way that the chemicals are bonded is by the electrons in that shell. Okay, there's a little bit of technicality there, but we don't really need to know that. But the most important thing to remember is that the electrons are what are joining together to make a chemical bond. Okay. So here's a very simple chemical: 1 carbon and 4 hydrogens. Okay. Ammonia, 1 nitrogen and 3 hydrogens, and water, H2O. Okay. Now the thing, or extra thing I want to find out here is notice how these different chemicals have different geometries. Okay. So this methane is a carbon with 4 evenly spaced hydrogens. This nitrogen has 3 evenly spaced hydrogens, and this water has 2 evenly spaced hydrogens, and these chemicals have a defined shape, okay? And how those shapes fit together within the cell is how we get a cell to actually be working. Here's a water molecule, and one of the things I want to point out here is that this oxygen, because the oxygen is negatively charged, that actually makes the oxygen partially negative and the 2 hydrogens partially positive. So that defines a polarity to this molecule, and most biological systems are going to have chemicals that are polar, and that allows them to sort of exist in the cell together. Okay. Another thing to think about is that water is the universal solvent. Okay, so you can have solvents or solutes. Solutes are the things that are dissolved in a solvent. The solvent is the liquid that things are dissolved into. Okay. Inside of a cell, and I think everyone gets this, but it's all an aqueous environment. Okay, so we have lots of water, and we can't survive without water in our cells. Okay, molecules that are within a microbe. All large molecules, those called the macromolecules, are made up of small parts. Okay, so for example, a protein is made up of multiple amino acids, with amino acids being that small part that can be combined into other parts to make the macromolecule of the protein. Macromolecules can always be broken down into simpler molecules. For this example I just gave, a protein can be broken down into the amino acids. Okay, so carbohydrates. So carbohydrates are one of the simple materials within a cell, and a major energy source within the cell. Okay, so carbohydrates are made up of carbon, hydrogen, and oxygen, and they're always in the pattern of CH2O. Okay, so there's 1 carbon, 1 oxygen, and 2 hydrogens for each part of the carbohydrate. And we can divide our carbohydrates into monosaccharides, disaccharides, and polysaccharides. So a monosaccharide is just 1 copy of the sugar. Disaccharide is 2 sugars linked together, and a polysaccharide is many sugars linked together. Okay. So simple sugars, double sugars, and many sugars. Okay. Monosaccharides are usually in between 3 and 7 carbons. So just to refresh again, if we had 3 carbons, we would have 3 oxygens and 6 hydrogens. Okay. When you have 5 sugars together, that is a pentose sugar, and the place we see those the most are in DNA and RNA. Okay. And the sort of go-to sugars are hexoses, and the major player there is glucose. Okay, and inside of a cell, everything breaks down to the glucose, and that's where we can, that's sort of our go-to monosaccharide. Okay. Maltose, again, 2 sugar rings, 1, 2, okay, that are linked together to make a disaccharide. Lactose is among the most prominent of the disaccharides. You know about lactose as being the sugar involved in milk. Okay. Sucrose, or table sugar, again, is a disaccharide. So it's 2 molecules of a glucose and a fructose to make sucrose. The way that we combine those sugars is through dehydration reactions. Okay, so when you're building them up, it's a dehydration synthesis. And one of the reasons it's called a dehydration is, you can see here we have 2 glucoses, okay, and each of them would have an OH at an appropriate spot, and you can remove the water, and that gives you the band between them, giving you the disaccharide of maltose. Okay, so to make a larger molecule, you do a dehydration synthesis. To split those 2 apart, you would do a dehydration reaction, and so then we would take this, get water back out of it, and give you 2 monosaccharides. Polysaccharides. So polysaccharides are when you have very large groups of sugars, and excuse me, most of these polysaccharides are used for energy storage. Okay, in bacteria, we can use some like a starch, or some. I'll have one other polysaccharide in a minute, but if you notice this chain of starch, okay, it's a whole bunch of repeating glucoses, okay, and that's how you get a starch. And just to try to relate it to some real world thing, you know that potatoes are quite starchy, and so someone with diabetes actually has a hard time eating potatoes, because you really don't want the pure glucose that you would get from a potato. Polysaccharides, besides being storage molecules, can also be structural. And in plants and algae, polysaccharides make up the cellulose. Okay, and especially in plants can be quite hard to break down. Okay. Now in bacteria, okay, we have a molecule called peptidoglycan, and this is the cell wall of the bacteria. And we'll definitely go over this more over the semester, but you have an alternating chain of two different simple sugars, and then cross bridges that are made up of peptides. Okay, just so I'm clear, it's not just any random sugar. Okay, these are very specific sugars that are in the cell wall, and hopefully I said that the peptidoglycan makes up the cell wall of the bacteria. With lipids, fats, phospholipids, and sterols, these are where we start getting into sort of more complex molecules, and they're of kind of various sizes. Okay, again, lipids are made up of carbon, hydrogen, and oxygen, but the big difference between a lipid and a carbohydrate is there aren't two hydrogens in this formula. Okay. Lipids are basically your fats and your oils. And one of the things that's very different for lipids is that they don't dissolve in water. So if you remember back to that picture of the oxygen, I showed you the partial positive and the partial negatives. A lipid will not have a partial charge on it, so it's nonpolar. And so because it's nonpolar, it can't dissolve in water. Phospholipids are what make up membranes. Okay, so they're a lipid that has some phosphorus to them. And sterols, one great example is cholesterol. Okay, and as much as people want to say, oh well, I don't want my cholesterol to go too high, that's true, you don't want it to go too high, but you also can't have cholesterol of 0. Okay, you need a little bit of cholesterol to make the cell walls more flexible. Now when we look at fats, you can see actually how we make up a fat. So we have a molecule of glycerol, and then we have these long chains of hydrocarbon. Okay, and you'll see again that we have some structural differences. So if it's just a pure chain of carbon and hydrogen, you have straight lines. Okay, you have a straight molecule. Or if you have a double bond in them, then you're going to have a bent molecule. And the degree of bent, or how many of these bent molecules are in a membrane, will also tell you how flexible the membrane is. Okay. Now, phospholipid is very similar, that we have these long chains of fatty acids, but attached to that glycerol, we have a phosphate group, okay? And one of the things that is sort of unique about this is that the phosphorus gives a negative charge to the molecule. So the head is always going to be facing outward of the membrane. Okay, the phospholipid head can't go, or the phosphate head can't go on the inside of a cell. sterols are going to stabilize the membranes in some of the microbes. Not that many, but in some of them. Okay. Cholesterol, like I said, is used in human cells. Our next major class of macromolecules are the proteins. Proteins are sets of amino acids and can also be called polypeptides because you join the amino acids together with a peptide bond. Okay. Proteins are made up of carbon, hydrogen, oxygen, and also can have nitrogen and sulfur in them. The vast majority of the molecules within the cell are going to be the proteins, okay? And these are really the things that do all the work within the cell. We can have proteins that are structural, giving shape to the cell, very, very important to transport things across the cell membrane, and also enzymes, or those proteins that do the work of breaking down things in the cell. Okay. So an amino acid is fairly simple. You have a carbon, okay, that has an carbonyl group on one side, the C double bond O, OH, and an amino group on the other side, the NH2, okay? Then every atom has to have four, or every carbon has to have four bonds to it. So this central carbon has a hydrogen and then some random group which could contain the nitrogen or the sulfur. Okay. There are 20 different amino acids, and those 20 amino acids can make up all the different proteins by putting them together in different orders. Okay. So here are some examples of the different amino acids, and again it's that R group, okay, the group here that's different among these. So we can have something, like a long chain of carbons, or possibly just one extra carbon and an H2. And the beta sheet. So these are the two secondary structures. And in a tertiary structure, this is the final folded form of that protein. And so you can see in this picture that these helixes or the beta sheets are used to build up a large three-dimensional structure. Now, every protein has to have a tertiary structure. Some proteins have a quaternary structure. And when you have a quaternary structure, that's when you have two different amino acids, or sorry, two different subunits that come together, okay? And not every protein has two subunits that come together, but some of them do. The major player that we think about like that is hemoglobin. Proteins are, again, totally dependent on shape, and the shape dictates what they can do. Okay, so if you denature a protein, you are unfolding those proteins. Okay, and heat is probably the easiest way to do it. And when you think about that, like for example, cooking meat, is giving you a denaturation. Besides heat, we can also use some chemicals. Acid is the major way that we do it. And so you can think about that in terms of the food ceviche. A lot of people will say, well, that's raw meat, generally fish, but there's generally a lot of vinegar in that. And so the meat is cooked because the proteins are being denatured by the acid. Microbes also have a nucleic acid, either DNA or RNA. Okay, all bacteria use DNA as their genetic material. Some viruses use DNA, some use RNA. Nucleic acids, again, are carbon, hydrogen, oxygen, nitrogen, and phosphorus. DNA is deoxyribonucleic acid, okay, and that is what most bacteria use as the storehouse for their genetic information, and RNA can be used to go from the DNA to make a protein. Okay. And one of the things we should remember when you're looking at DNA is that you have a pentose sugar, okay, and then a base that comes off of that, and then these are made into large chains with these phosphate groups, okay. Now the difference between DNA and RNA is the sugar, okay. So in deoxyribonucleic acid, you have no oxygen here, okay, whereas in RNA, you do have an oxygen here. And so that is defining the difference between DNA and RNA. Okay. When something is made into a nucleotide, it has a phosphate group. And again, to make a long chain, we're going to do a dehydration synthesis again, so that again we're going to lose water, and it's going to be linked through the through the phosphate groups. Now, the bases A, T, C, and G in DNA, and you have uracil in RNA. Okay, so the bases are very similar, and like I said, you have something like A and T, and so there's always a 2 ring structure and a 1 ring structure that link together to make the base pairs. Okay. When you're looking at the base pairs, you're always going to have that the number of A's and the number of T's are always going to be the same, and the number of C's and G's are always going to be the same in DNA. Here's a model of the double helix, showing you that the sugar phosphate backbone is on the outside, and then the bases are across from each other to make up the double helix. Okay, so there's a few final thoughts. That all living things are based on atoms. the basic chemicals that are inside of cell are carbohydrates, lipids, proteins, and nucleic acids. A thought question for you, could you eliminate one type of macromolecule without affecting life as we know it? And yeah, I'll let you think about that.